based in the transcriptomic profile of the receptive endometrium. We have developed this test by analyzing RNA expression from women in different moments of their menstrual cycles to define the transcriptome of the receptive endometrium. So now, every time that we analyze the transcriptome of a patient, we can classify this endometrium whether it's receptive or pre-receptive or post-receptive. And this is because the window of implantation is supposed to be in a concrete time of the menstrual cycle. For example, in a HRT cycle, it should be around E+5 days after progesterone administration. But in some women, we have this window of implantation is a little bit delayed or a little bit advanced. So if we take the standard practice to transfer blastocyst in P+5, for some women it will work, but some other blastocysts will not have the chance to implant because they were not in the right place in the right moment. So we believe that it's important to synchronize the embryonic development with the endometrium so that we can transfer the blastocyst in the moment that is going to have more chances to implant. And using this criteria, we have been performing this test in many patients, 20,000 but more now these days. And we can find that nearly 30% of the women that we analyze have a receptive window that is displaced, so only changing the days of administration of progesterone can improve the rate of this patient. But still doing this, that is a very nice test and that is working very well, we found some women that even having the best embryo ever and the best endometrial, the personalized embryo transfer at the time of a window of implantation, these women don't get pregnant. So we were wondering whether we can find other factors that can play a role in fertility and of course in improving ART. And we came to start working with microbiome. And why do we start microbiome? But first of all, because the technological advances has allowed us to analyze the bacterial composition at a very, very precise level and in low biomass microbiomes that we thought before that were sterile niches. And this is a slide that I really like because this is very astonishing information about the microbiome. Nowadays we know that we have in our bodies 10 times more bacterial cells than human cells, which is a lot. And that from these 10 times more bacterial cells, they contribute to 90% of the genes that are expressed in our body, mainly in the gut, but in many other places, the skin, the airways, and everywhere. And that nearly 2% of our body weights belongs to bacteria, which is also very impressive data. So we started working with the human microbiome and we focus in the urogenital tract. This is information that was published by the Human Microbiome Project. This is a project that has analyzed nearly 400 subjects, healthy subjects, and they have reported the different microbiomes that you should expect from different body sites. If we focus in the urogenital tract of females, we see that, as Professor Ventolini has already explained, it's mainly composed of lactobacilli, but you can also find some other bacteria that we can consider dysbiotics, like for example Prevotella, Barnarella, Atopobius, Neatia, and so on. And what do we know about vaginal microbiome and infertility? So we know that up to 40% of patients that attend an IVF clinic, they present abnormal vaginal microbiota. And also we know that bacterial vaginosis is responsible for a higher risk of miscarriage, premature rupture of membranes, and also preterm labor. So we started to think about the endometrium because we think that this is not only located in the vagina, but following this slide that I like to borrow from Frank and Scott, we think that this is something that happens. This microbiome is involved in the whole process of reproduction, from the production of gametes up to through the pregnancy and until the baby is born. So we ask ourselves what is happening in the endometrium, which is the interface in which the blastocyst meets the mother and they have to interact and the embryo has to adhere. So we started looking at the literature and we find different papers, and we selected, I have selected these 6 papers that were very interesting to us. In the 6 papers, they use more or less the same design, but these in IVF patients in which they were going to do the embryo transfer. At the time of embryo transfer and after transferring the embryo, they cut the distal tip of the transfer catheter and they put it in culture, and they culture different microbes. At this time, it was the sequencing, the deep sequencing was not very in the market, so they did this by microbial culture. And what they found is that every time, in every patient that they isolated these pathogens that you can see here, Escherichia coli, Klebsiella, Staphylococcus, Streptococcus, and different enterobacteria, they found that the implantation rates and the pregnancy rates of these patients were lower compared to those that did not present these pathogens. And the other way around, the miscarriage rate were higher in these patients that presented these pathogens in their endometrium at the time of embryo transfer. So with this evidence, we prepared a study that the results of the study were published in 2016 in the American Journal with 2 main objectives. The first one, to test the existence of an endometrial microbiome, and the second one, to analyze if this endometrial microbiome can play a role or can have an impact on reproductive outcome in IVF patients. So we used this method. What we did is we took the sample, in this case endometrial fluid or vaginal aspiration. We obtained the total DNA, and taking advantage of this gene, 16S ribosomal RNA gene, this is a very interesting gene for metagenomics because it presents 9 different hypervariable regions that are specific for each bacterial species. So if we know the concrete nucleotide sequence, we can assign a taxonomy to these samples, and then we can have like a relative abundance of each bacteria in this sample. So using this method, we designed a first pilot study in which we analyzed healthy and fertile subjects in their natural cycles, and we took the samples in the pro-receptive and in the receptive states, and comparing endometrial fluid and vaginal aspirate. So 2 samples in the same woman in the same time. And these are the results. This is a summary of the results of what we found. We found that in the endometrial fluid and vaginal aspirate of this woman, really, the microbiome found in the different two places, it was very similar, but it was not identical. We found some specific bacteria that were only found in the endometrial fluid. Also, if we compare these samples in pair samples of endometrial and vagina taken from the same woman at the same time, we see that many of them are very, very similar, but there were some women that presented, for example, this one here, the first one, presented different microbiome between the endometrium and the vagina. So this was the first evidence that the endometrium can have a own microbiota. So for the second objective, we started working with IVF patients. In this design, we studied 35 patients that came, all of them were patients with repetitive implantation failure. And what we did is, in the same day that we are taking the endometrial biopsy for the ERA test, endometrial receptivity analysis, we took, just before taking the biopsy, we took some endometrial fluid. So we analyzed the microbiota in the endometrial fluid, we did the ERA test in the endometrial biopsy, and we correlated the microbiota with the receptive status of the endometrium. Then in those patients that were receptive, they went for embryo transfer, and we correlated this microbiota with implantation and later on with the pregnancy outcomes. And when we did this test, this was the first picture that we saw of our results. And this is a heat map, and what we can see here is in each row of this graph, you can see bacterial genus, and I have represented here the 20 most abundant in the study, and each row regards to all of the patients that we studied. And you can see that there are, first thing that you can see is that there are some bacteria that are very enriched in a population of women. And these bacteria, the most prevalent were Lactobacilli, Streptococcus, Bifidobacterium, and Gardnerella. So we ask ourselves whether we can predict the pregnancy outcomes of these patients just looking at the amount of these bacteria in their endometrial fluid. So we use two different models to predict two target classes. First one, it was a successful outcome, which is live birth. Second one was a failed outcome, which is no live birth. And we have included in this last group the patients that either didn't get pregnant or those that got pregnant but then had a miscarriage. So we have four variables, two target classes, and we use two different mathematical methods to try to predict this. And we use in the one hand classification and regression T, and in the second hand we have a generalized linear model. And both mathematical models presented finally the same result. That was the percentage of lactobacilli was able to predict the success or the failure of the infertility treatment. And both models presented a cutoff value of 90% of lactobacilli. So if we come back to our heat map and we can classify our patients in two different groups. The first group is the one in the middle with more than 90% of lactobacilli, and we call this group lactobacillus-dominated microbiota. On the other side, we have these other patients that had less than 90% of lactobacillus in their endometrial fluid, and we call this group non-lactobacillus-dominated microbiota. So based on this classification, we group our patients. We have here the number of the patients, the subjects, the outcome, and we analyze, this is the composition of the bacterial microbiota, and we classify them into LD or NLD, depending on this 90% of lactobacilli. And this is difficult to see here, but if we see the average numbers, we see that patients that had a live birth presented a higher amount of lactobacilli, which is represented in blue, than those with a miscarriage or non-pregnancy. And also that those women who had a miscarriage or did not get pregnant, they presented a higher abundance of Gardnerella vaginalis, which is here in red, or Streptococcus, for example. So this was also confirmed using other methods of analysis. In this graph here, we see that lactobacillus is associated with successful treatments, while Gardnerella, Bifidobacteria, Helicobacteria, Clostridia are associated with failed cycles. So these are the statistics. If we compare the group of women having a lactobacillus-dominated microbiota and those with non-lactobacillus-dominated microbiota, we see here that the implantation rate, the pregnancy rate, the ongoing pregnancy rate, and the live birth rate per transfer were higher in the group with lactobacillus-dominated microbiota than those with a dysbiotic or abnormal endometrial microbiota, and the other way around, miscarriage rate were higher in this with non-lactobacillus-dominated microbiota, and this difference was not statistically significant. But again, the numbers are still very low, so we cannot conclude for this outcome. So the summary of findings up to here is that we support the concept that the uterine cavity is not a sterile site, that although the endometrial and vaginal microbiota are very close related in healthy and fertile women, you can find differences in the structure and composition between them, and they are observed in approximately 20% of the women. So this shows that these two sites are not identical in every woman, so maybe it's worth to analyze them carefully. Also that the endometrial microbiota profile can be classified as lactobacillus-dominated or non-lactobacillus-dominated, and this classification enables the diagnosis of endometrial microbiological health of IVF patients, and also associates with their reproductive outcome. And also that a non-lactobacillus-dominated microbiota, especially when you find Gardnerella or Streptococcus in these samples, strongly correlates with adverse reproductive outcomes when compared to subjects with lactobacillus-dominated microbiota. So, well, this is, I've talked up to here is the results that we have presented in these papers, and I would like to show you a little bit of what we are going to do in the future, what we are doing in fact now. We are doing this prospective study to validate these results. As I said before, this was a pilot study with 35 patients So we are doing now a prospective study, multi-center international, with approximately 400 patients worldwide. And we are going to analyze this endometrial microbiota in ART patients with indication of endometrial receptivity test, because they are patients with recurrent implantation failure, and that are going to transfer a blastocyst, frozen blastocyst in day 5 or 6. So this is the participant sites worldwide. We have selected patients all over the world because there are some papers that have demonstrated that at least in the vagina, the microbiome is different among different ethnicities. For example, Afro-American women can present what we said this is an abnormal microbiota, and they have no symptoms and they have no problems with infertility. So we have selected from Asian patients to different clinics, different ways of working, and different ethnicities, of course. And we are very happy that some months ago we found this paper in Nature Communications, and in this paper they have designed a very elegant work in which they not only analyze the endometrium and the vagina, but they complete the full picture of the reproductive tract. In this paper, they analyze from the vagina to the cervix, endometrium, also the ovaries, the tubes, and even the peritoneal cavity, and they have found results that are consistent with what was published before by our group and also others. And they have also related this this microbiota in the reproductive tract with not only infertility but also with endometriosis and also some types of gynecological cancers. So this is giving more importance, more relevance to the microbiota in this field. So the conclusions are that the existence of a non-lactobacillus bacteria in the endometrium negatively impacts the reproductive function and should be considered an emerging cause of implantation failure and pregnancy loss. And the results that are presented here expand the evaluation of the endometrial receptivity not only at the morphological and molecular levels, but also at the microbiological point of view. And this is how we foresee the future. For us, a possible way to improve these IoT outcomes would be to analyze in the endometrium not only the receptive status of this woman at this time using the ERA test or some other predictor of endometrial receptivity. So in the same sample, we would like to analyze the human RNA to say which is the endometrial status of the endometrium, but in addition to analyze the bacterial DNA so that we can complete this information by saying you are lactobacillus dominant or not, and you are in the best momentum for implantation or not. And in the last minutes, I would like to show you this work. This is a side project that we had when we started working with endometrial microbiota, and it's about chronic endometritis. As you know, the chronic endometritis is persistent inflammation of the endometrial mucosa, and it's mainly caused by different bacterial pathogens, for example, Staphylococcus, Enterococcus, Enterobacteria, Gardnerella vaginalis, Ureaplasma, Mycoplasmas, and so on. So this is, for us, this is the best model of an abnormal endometrial microbiota. So we wonder whether we can improve the diagnosis and the treatment of chronic endometritis. So for this study, we contacted Professor Ciccinelli from the University of Bari. He has been working for a long time with chronic endometritis, and he has developed, apart from publishing many papers, he has developed this hysteroscopy method to diagnose chronic endometritis. So the design of this work, Professor Ciccinelli was taking endometrial biopsies at his office for a set of patients, and we blindly analyzed 4 different tests to diagnose chronic endometritis. We were doing, at iGenomics, we were doing molecular diagnosis, either using RT-PCR or 16S sequencing, and in his part he was doing histology, hysteroscopy, and microbiology. all the analyses were blind for all the participants. At the end of the study, when we had all the samples analyzed, we put everything on the table and we started to see which was the concordance between the different methods. And we were very surprised to see that we had every different case. Starting with, these are some examples, but I can show you some more. This is, for example, this patient here who has a negative histology, negative hysteroscopy, but then the microbial culture was positive. She was also positive for RT-PCR, for the molecular methods, but then microbial culture and RT-PCR were discordant, so it's difficult to say to this patient if she is positive for CE or not, so it's a little bit confusing. Second case here, negative for histology, positive for hysteroscopy, and negative for microbial culture and molecular methods. But we also find some other cases in which patient was negative for histology, was positive for hysteroscopy, then the microbial culture was positive and it was confirmed by molecular methods. So in this case, it was, for me, it's quite clear that she has streptococcus in her endometrium, but still the histology, which is nowadays the gold standard, was negative. So how can you manage with these patients when you have different evidences of having or not chronic endometritis? So I have summarized these results in this very simple graph here. If we compare the three classical methods, I'm not talking now about the molecular microbiology, just histology, hysteroscopy, and culture, we compare these 65 patients in which we had results for the three classical methods. And as you can see here, the accuracy of the method was 20%. Only 13 patients out of these 65 patients presented the same result when the sample was analyzed in 3 different ways, which is very, very low to what we expected at the beginning. And normally, what we found is that hysteroscopy usually overdiagnosed chronic endometritis, while histology sometimes underdiagnosed chronic endometritis. And at the end, the only way to know which was the causing pathogen was doing the culture. So which is the method that we should use to identify this disease or not? So what we have done, well, the paper was accepted 2 weeks ago, and I think it's now in Moreno, in a couple of weeks you can see the whole work. But what we have done is to analyze all these patients, but specifically these 13 patients that had concordant results for the 3 classical methods using this molecular microbiology. And we found that from these 13 patients that were concordant for the 3 classical methods, we were able to obtain the same results in 10 out of these 13. So it's nearly 77% of the women analyzed. So in conclusion, these are the index texts: sensitivity, specificity of 75 and 100% of molecular microbiology compared to the classical methods, positive and negative predictive values 100% and 25, false positive rate 0%, false negative rate 25%, and accuracy, as I said before, 77%. So what we propose is that molecular diagnosis of chronic endometritis is similar to using histology, hysteroscopy, and microbial culture together, and maybe can overcome the bias of using any one of them alone. So we propose that chronic endometritis as an infection disease has to be analyzed and to manage the patients and the fertile patients. So last thing, I would like to thank Professor Carlos, which is the scientific director of Fertility, for allowing me to do this research. All the research department, especially to Marta, Davide, and Yolanda, who are the microbiome team, and of course our collaborators, Professor Cicinelli, but also Pilar Alamar and Sergio Cabanillas from ISGE Valencia, because they were recruiting the patients and collecting the samples, and without their help, this work is impossible. Of course, all the iGenomics team, and thank you very much for your attention.